We're back with lecture 6, the second part of it in our statistics series. In our last lecture, we introduced the idea of null hypothesis significance testing, the pros and cons, different ways that we can use it. And we ended the last lecture with the idea that null hypothesis significance testing can be a very helpful measure to report, but we need to report it in conjunction with some other things. Those things that we talked about were, if we remember, power calculations, effect sizes, and some other things, but those were the two we talked about. So let's define some concepts of things that are really, really helpful to report in addition to our hypothesis testing. So let's start with effect size first. So an effect size statistic is basically a standardized measure of the size of the effect of a variable, or in other words, the strength of the relationship between two variables. So here it says a measure of the magnitude or meaningfulness of a treatment effect or of the relationship between two variables, independent of the sample size. The effect size and sample size, sometimes people get confused because they have the same word size in it. Sample size is the number of participants. Effect size is the meaningfulness of the relationship that you find. We can also interpret it as an indication of the practical significance of your research finding. And effect size values should always be reported along with your p-value and should always be considered when you are interpreting results of your own studies and of other studies that you're reading. Your p-value is going to tell you whether or not an effect was found, but the effect size tell us how important or substantial that effect actually was, which is why it's important to report them together. So here is how we measure effect size. There are a couple of ways that we can measure it, but one of the most common is called Cohen's d. It's a measure of the effect size that indicates the number of standard deviation units by which two means differ. So similarly to some other concepts that we've talked about, like a z-score or something like that, Cohen's d is in units of standard deviations, and you find it by finding the difference between two means, two population means, and then you divide it by the standard deviation. Here are the effect size conventions. Generally, if you have an effect size that's 0.2 or lower, that's going to be a small effect size. Well, at least 0.2 is a small effect. Anything under that, we don't really count. 0.5 is going to be a medium effect size, and then 0.8 or larger is going to be a large effect size. Let's talk about how this applies to research. So for example, if we imagine that there's a new treatment for older adults who have memory problems, because the treatment works, your treated population performs better than the untreated population on memory tests, hypothetically. The magnitude of the difference between the population means pre and post treatment, right, is called your effect size, and that's what we'd put up here. Let's say, hypothetically, that the memory test scores have a population mean of 100 and a standard deviation of 15 in your untreated population. And then in the treated population, the standard deviation is still 15, but the mean is going to be 106 instead of just 100. The two means are going to differ by 6 points, so it would be 6 over 15, which is equal to 0.4. So that would be 0.4 standard deviations difference between the mean in both populations. So your effect size 0.4 is going to be a small to medium effect size. It doesn't quite reach the threshold for medium, so you would still say small. But that is how you would find the effect size. The next thing we're going to report with our null hypothesis significance testing is called confidence intervals. So confidence intervals are, as you see up here, a range of values that is estimated from sample statistics with a given probability that the range will contain the true value of the population parameter. In other words, if we were hypothetically looking for the mean, we find a mean in our sample, or using our sample statistics, we are going to create a range. For example, let's say like from 85 to 95. We're going to say we have a confidence interval, a 95% confidence interval from 85 to 95. And what that means is that, I mean, you can interpret it as we are 95% certain that the true mean of the population falls between 85 and 95 as a hypothetical. We're going to talk, that's not really what a confidence interval defines, but that's often how we see it used. And we're going to get into that in a second. But some notes are that the width of the confidence interval, how large it is going to provide information about the precision of the estimate. So the wider or bigger your interval is, the farther apart those, those two numbers are, is going to say that you have relatively low precision of your estimate. But if you have a narrower interval, the numbers of your range are closer together. That means you're going to have a more precise estimate of what that true population parameter is. These bullets are important here too. It says it provides an estimated range of values within which the actual population mean or parameter is believed to fall. But the other thing to know is we can use it to tell if we have significant findings. If our confidence interval contains 0 somewhere in the interval, you cannot claim significance. But if your interval does not include 0, then you can say you have a statistically significant finding. And we'll get more into that later in some of our later lectures. The precise definition of what a confidence interval is, I'm trying to decide if I want to get into this now or not, but I'll give you a sneak peek. A 95% confidence interval of the mean, calculated from a sample, implies that if the samples originate from the same population, you're drawing all of your samples from the same population with the same extraction method, random sampling, hopefully, 95% of their confidence intervals that you find from those samples are going to contain the population mean, the true population mean. For example, if we calculated a 95% confidence interval of the mean from our data, and we repeated the same experiment 100 times with 100 different samples, 95% of those samples would give us confidence intervals that contain the actual population mean. Five of those would give us a confidence interval that don't actually contain the population mean. This differs slightly from the explanation that a 95% confidence interval of the mean calculated from a single sample includes the population mean with a probability of 95%. They're slightly different, but often you can use them kind of interchangeably when you're reporting on your own data. And these are also very important to report with your p-values. The last thing we're going to talk about is it's important to state the statistical power of your data, your experiment. Power, we've talked about before, 1 minus beta, is the probability that the test will correctly reject a false null hypothesis, or in other words, that the probability that a test will correctly identify a treatment effect when one actually exists. We talked about this a little bit in our last lecture. Researchers are going to typically establish the power that they want their experiment to have through something called a power analysis before they actually even start conducting the research. This is a kind of a way of setting yourself up for success and determining different variables that you need to establish within your research study to make sure that it is successful and that it gives you the data that you want. Typically, studies, as you can see here, aim to achieve about 80% power, which is a 20% risk of a type 2 error, which is a false negative. But that's just generally. But there are a couple of factors that you're going to be able to change to increase your power if you want to. Those are increasing your alpha, having a larger effect size, increasing your sample size, and then changing from a two-tailed to a one-tailed test in the correct direction. In the next couple of slides, we're going to look at each of these relationships. We're going to play with it and reason through why each of these relationships are the way that they are, because not all of these are created equal. Some of these are going to be things you're going to want to do to increase power, and some of them are things you're not going to want to do. The first thing that we should see here is that increasing our alpha increases our power. If we increase our alpha, let's wait for it to turn back around. If we increase our alpha, which is, remember, the probability of that we'll get a type 1 error, increase the alpha, your power also increases. But this is generally a bad idea because by definition, as we're increasing alpha, we increase the risk that we're going to get a false positive, right? We're going to say that there's a treatment effect when there actually isn't one. Which is why your alpha level should be determined before you even start these power analyses, because if it's because you don't want to mess with it in order to increase your power. Although it's your prerogative in your own research to set your alpha to whatever value you want it to, it's also everyone else's prerogative to distrust your research findings if your alpha is set too high. It's generally recommended that you keep your alpha at the standard level for whatever is common in your field of study, which in most cases is just 0.05. But as you can see in this animation, decreasing your alpha is going to decrease your power, and increasing alpha is going to increase your power. But you can also see the problem with lowering your alpha too low. If you have your alpha too low, then your power is also going to be really low, which is not a good thing. So you need to find a balance between having a reasonable alpha level that people will trust your research, but then also having a reasonable power. It's not a linear relationship, which is why there are other things that we're going to do to increase the power of our studies if we want to. We're not going to really touch our alpha. As the other relationship is, as our effect size, or the difference between our two means here, as the effect size increases, our power also increases, which is the probability. In general, what this means, though, is that the probability, as your effect size increases, the probability of rejecting the null hypothesis increases, which means that your power of your test, it also increases. And that makes sense, right? When you have a large effect size, that means that the change between the means is going to be easy to detect, aka high power. If you have a small effect size, the change between your means is going to be a lot harder to detect, aka lower power. Again, effect size isn't really something you can change. It's just something that either your experiment, well, there are things you can do to adjust effect size, but a lot of times it just depends on what treatment you're looking for. So again, alpha, effect size aren't really things we want to change when we're looking at increasing power. This, however, is something we can change when we're looking at increasing power, increasing our sample size. As you increase your sample size here, what that does is it decreases your standard error, which makes the mean of your sampling distribution narrower, which just decreases the overlap here, which decreases your type 2 error rate, which then also increases your power. This is something that is actually going to be a very helpful strategy for when you want to increase power. One of the main reasons that we do a power analysis before we start a study is to figure out what sample size we need to achieve a reasonable probability of a successful research study. So that's one of the things that we look at. What sample size do we have to achieve to get the power that we want? And remember, we said that a general good power to shoot for is 80% or 0.8. Okay, and the last thing that can increase power is switching from a two-tailed test here to a one-tailed test. That increases your power. However, one-tailed tests only have increased power, increased probability of correctly rejecting a null hypothesis or correctly identifying a treatment effect if you put the tail in the right direction. If you hypothesize that you're going to have a positive effect, and so you put your tail on that side, but it turns out that you actually have a negative effect, your power is actually going to be 0 because there's no way to essentially reject the null hypothesis because you don't have any data pointing that direction. So because of this, because you really don't want to risk accidentally setting your tail on the wrong side, most researchers just opt to have a two-tailed test instead. Even though they're less powerful, They prevent the awkwardness of having to retain the null hypothesis, even when your mean difference is huge, just in the wrong direction. But this is something that can increase your power if you are pretty dang confident that your effect is going to be in one direction or the other. So after we've talked about all of these things, let's go through some best practices when we're using hypothesis testing. So the first thing we have to do is justify our use of a null hypothesis statistic test. Remember, it's designed to be a heuristic. It's not something that's designed to It's not something that is designed to establish the next scientific finding or fact. It's just something that we use to figure out if it's worth it to pursue further research. Heuristic. The next thing we need to do when we've decided that we want to use this investigatively is we need to establish the power and the alpha of our test a priori, which means before we start collecting data, before we start setting up the experiment, things like that. Another thing we can do is pre-register our study to a site like osfio.io, which is just somewhere where you can put your research plan, your research, your analytical methods, things like that, where other researchers can comment on your ideas and give you some feedback. It holds you accountable, things like that. Remember also that non-significant findings, not when you accept the null hypothesis, are just as important as significant findings. The idea that people only publish positive results when we see a change leads to something called the file drawer effect. This just means that research that doesn't support a researcher's hypothesis gets filed into the drawer and not published. Nobody sees it, which can lead to extremely biased science that is published. We need science that says we didn't find an effect just as much as we need papers that say we did find one. Because otherwise, how else are we supposed to know which direction to take science in? So publish your non-significant findings. Another thing to do with hypothesis testing is to publish your raw data so that other people, if they are interested, can also look at what you did for statistical testing, compare it to the results that you got, things like that. We need to report effect sizes and confidence intervals like we've talked about today, and also phase out statistics light education, which we hope is something that we are trying to combat with this course. Statistics light education is just the idea that we only know how to plug things into formulas. We don't know why we're doing it or what it means. With this course, you will know what every single one of your statistical test means and why you're running it, hopefully. Before we finish up, let's look once again at the APA style for reporting results of hypothesis testing. When you're reporting the results in your manuscript, you don't state that you reject or you fail to reject the null, but you write your results like this. So as expected, which means that it's telling which hypothesis you're confirming, the sample of women with lifelong depression were shorter on average, and then you give the mean, than the general population, and then the population mean. A two-tailed one-sample z-test with an alpha of 0.05, this is your methods, demonstrated that this difference was statistically significant, and then you're reporting your critical value, or the z statistic that you got, and also your p-value. In addition to this, if you were to write this, you would also need to report, remember, your effect size and your confidence interval. Note that all your M, Z, and P, your sample statistics, are all printed in italics. The Greek letters for your population are the only things that should not be in italics. It's necessary to state which statistical test you used, the alpha level, the number of tails, all of that prior to reporting your results. You usually do this in your methods section. Sometimes you'll have a specific section in your methods for statistical analysis. Usually you will, actually. And APA requires that significant p-values be written out exactly to the second or third decimal place with no zero before the decimal point. So just like this, 0.008, not just p is less than 0.05. But when it is less than 0.001, just write p is less than 0.001. And here is an example of it in the literature. You're welcome to take a look at this, see how it's worded, and see if you can find what type of test they used, the significance or the effect size, things like that. See if you can figure that out from this results section. Okay, and with that, we're going to end this lecture, and I will see you again in your next lecture, where we explore some more different aspects of different types of statistics that we can use other than the Z statistic. Okay, and I will see you in the next lecture.